At 8:17 p.m., a death record can contain one official time, but inside the body, no master clock reads 8:17. Temperature follows one curve. Blood follows gravity. Muscles follow their chemical energy. Cells and microbes follow other schedules. That is why the first 24 hours after death do not unfold like a television countdown. The first surprise happens almost immediately. The body does not become stiff. It relaxes. And although the person has died, every cell does not stop in the same instant. Death is one boundary for the human being, but several different endings for the body's parts. This timeline begins after death has been medically determined, not merely when a pulse disappears. Cardiac arrest can sometimes be reversed, so clinicians use accepted criteria appropriate to the situation and jurisdiction. From here, assume circulation has permanently stopped. This is not about the final conscious seconds or an afterlife. It is the measurable biology that begins once blood no longer carries oxygen through the body. In the first minutes, the skin may grow pale and the muscles lose the low-level tension they had in life. This is primary flaccidity. The jaw and limbs relax. Movies often reverse the sequence by making a body instantly rigid, but rigor mortis comes later. Microscopically, cells use their remaining fuel as energy stores fall and membranes become less stable. Different tissues tolerate the loss of circulation for different lengths of time. That does not mean the body is recoverable or awareness remains. It means the death of the organism and the death of every individual cell are not the same event. The temperature clock also starts, but it is a terrible stopwatch by itself. The body gradually approaches the temperature of its surroundings on a curve that can lag at first, fall faster, and then slow again near room temperature. Body size, clothing, moving air, water, contact with a cold surface, fever, and ambient temperature can all change that curve. So the television rule that a body loses a fixed number of degrees every hour is only shorthand. Real forensic models need several correction factors, and even then they produce an estimate, not an exact timestamp. While heat moves outward, blood moves downward. With no heartbeat keeping it in circulation, gravity draws blood toward the lowest parts of the body while it remains inside the vessels. This produces livor mortis, or lividity. It can begin appearing within the first hour and usually becomes clearer over the next several hours. Areas pressed against a firm surface may remain lighter. Early lividity can partly shift if the body moves. Later it becomes progressively less mobile. But there is no universal minute when it becomes fixed. Temperature, blood volume, pressure, and other conditions affect the pattern. Lividity is a clue with a range, not a clock with a minute hand. Then the muscles reverse direction. Muscle fibers contain protein filaments that grip each other to contract. In life, a molecule called ATP supplies the energy needed to release that grip. After circulation stops, cells can no longer keep making ATP normally. Once the remaining supply falls far enough, the links cannot separate, and the muscles stiffen. Rigor is often first noticeable around the eyelids and jaw within the first few hours and becomes easier to detect elsewhere later. It may be broadly established around 6 to 12 hours, but those are guidelines, not appointments. Temperature, illness, age, body condition, and activity before death can alter the pace. By 24 hours, rigor may still be strong, while warm conditions may already be helping break it down. The strangest clock operates at the cellular level. A study using more than 7,000 samples from 36 human tissue types found that RNA and gene expression patterns changed on different schedules across roughly the first day. In blood, many of the largest measured differences appeared between about 7 and 14 hours compared with samples taken before death. Some changes reflected short-lived cellular responses to oxygen loss and stress, not random decay alone. Another study obtained thin slices of human brain tissue during autopsies performed within 8 hours of death. Under carefully supplied laboratory conditions, some neurons and supporting cells could be maintained for weeks. But this is not a functioning brain or hidden awareness. One house running on a backup battery does not mean the city is alive. These findings show cellular resilience, not consciousness, recovery, or proof of an afterlife. Meanwhile, autolysis, literally self-digestion, has begun. As cell membranes fail, internal enzymes start breaking down nearby structures. The barriers and immune defenses that kept microbial communities in place are also gone, allowing microbes to contribute to internal change. Warmth and humidity accelerate these processes, cold and refrigeration slow them. Visible decomposition may begin within a day in some warm conditions but take much longer in cooler settings. For much of the first day, the most important changes remain microscopic and internal. That is why real investigators do not solve time of death by touching the skin once. They combine core and room temperature, lividity, rigor, the scene, clothing, body size, medical history, and records of the person's last known activity. The clues can disagree because a heater switched on, a window opened, the body moved, or the starting temperature was unusual. That disagreement is not a failure. It proves the clocks experienced different conditions. After 24 hours, the body has relaxed, cooled, settled, stiffened, and begun changing internally, but not on one universal schedule. One official time can mark when a life ended. Inside the body, the clocks never agreed, which is why honest forensic science gives a range instead of a perfect minute.